Okay, polymerase chain reaction. We have mentioned PCR in this class before when we talked about DNA sequencing. We know what the ultimate goal is, but now we're going to go into how PCR actually occurs. So, the goal of PCR is to amplify DNA or make more copies of DNA. Now, the key thing is we're not trying to amplify the entire sequence. Okay, the whole genome for a human has 3 billion base pairs. That's a really big piece of DNA. So with PCR, we only want to amplify certain segments depending on what we are searching for. So we only want to do a target sequence and that's it. And we're going to amplify that target sequence. Now, the first step is we have to add supplies to our PCR tube. Okay, and the key things you have to have in there. First one is your DNA sequence, your target DNA sequence. Okay, we've talked about how to isolate out DNA, and we are going to do this on Monday. So we get our DNA sequence in there. Then we add our primers. Primers are starting points for that DNA sequence because we can't replicate DNA unless we have a starting point. And so those primers are our starting point. We're going to have free nucleotides in there because we're going to be building new DNA strands. So we have to have our adenine, guanine, cytosine, thymine. They're just going to be floating around in that mixture, and they're eventually going to get used to make DNA strands. The last thing is Taq polymerase. Now, Taq polymerase is an enzyme, ends in ACE, so that's how you know it's an enzyme, and its job is to build new DNA strands. Now, the key thing about Taq polymerase, it's actually a little special little guy because Taq polymerase is able to withstand extreme temperature changes because most enzymes are very sensitive to temperature changes. And so Taq polymerase is great because it's like, yeah, I'm fine. I don't have to worry about it. I'm good. I don't have to worry about extreme temperature changes. And so that's why we use Taq polymerase instead of the normal DNA polymerase that is what you use in your cells when your cells replicate. So we get all these supplies, we put them into a PCR tube, and it's just going to look like a clear liquid. You won't see any of the individual pieces, but we are going to hope to see the final results. So once we have everything in the PCR tube, we put it into what's called a thermocycler. Thermocycler is a machine that is able to control the temperature. By controlling the temperature, it's able to make it hot, cold, in the middle, hot, cold, in the middle, and it basically just repeats over and over and over again. And so if you were to stick your hand on top of the thermocycler, you can actually feel the temperature changes. It's like a really good oven. Okay, it just changes temperature very quickly so that we can accomplish our goal. So with the thermocycler, it has 3 major stages. The first stage is it's going to heat things up to 94 degrees Celsius, which is hot. Okay, so we're going to get it hot first. And when we get it hot, it denatures the DNA. So we know DNA is double-stranded, so it's going to separate out those 2 strands. So it's going to break them apart. That's what denature means. Then we're going to cool it down to 55. So we're going to cool it down. We get it hot, and then we're going to cool it down. When we cool it down, those primers, which are those starting points for our sequence, those primers are going to attach. And when the primers attach, that is our starting point. Then we got to warm it up a little bit. So we go from hot to cold, then we got to get a little bit warmer. Okay, when we warm it up, that's when Taq polymerase, it says, okay, I'm ready to go. And Taq polymerase starts adding on those new nucleotides. Then we repeat. We now have new DNA strands, so we separate all of those. We denature them in the hot. Then we're going to put on the primers. So we anneal, A-N-N-E-A-L is anneal. So we cool it down and we anneal. Then we warm it back up and we extend. We add more DNA nucleotides. Then we start over again. So we heat it up and all of those new strands of DNA all separate. Then we cool it down and the primers glue or they anneal. So the primer is set down. Those are our starting points. Then we warm it back up and Taq polymerase comes in and it adds the new DNA strands. Okay, that is the overall process in the thermocycler, and it just repeats over and over and over again. Now, the end result is you can program your thermocycler to do as many cycles as you need. So we're going to do a lab on Monday where we're going to have it do 10 cycles, then we're going to have it do 20 cycles, then we're going to have it do 30 cycles. The more cycles you do, the more DNA copies you end up with. So by doing 45 cycles, you end up with millions of copies of your target DNA sequence, just because we break it apart, we glue down the primers, then we extend new DNA, and then we repeat over and over and over until we have millions of copies. Now, I listed some videos where at the same place you got this video. I want you to go in and watch them, because really this works whenever you see the animations and you see it occurring. But think about those steps of PCR as you go through. Denature, anneal, extend. Denature, anneal, extend. Okay, that's the key thing to take away. You do have to know the temperatures. You do have to know how it works. Good luck.